All right, this video is on biochemistry. So similar to, there's also a video that we have on the cell. It goes over quite a few concepts, but really not in a ton of depth. So a lot of this you may already be familiar with, but biochemistry questions are on the AP Bio exam, and so it's important for us to go over this stuff. Okay, so we have 5 questions here, and these questions will show up again at the end of the video, at which point it's a good idea to try to answer them for yourself. What is an isomer? A metal bonded to a nonmetal. What is that? The pH scale and what makes up acidic, neutral, and basic pH numbers. What's an isotope? And which law states that entropy of any isolated system always increases? All right, so it's basically the laws of thermodynamics. You need to be familiar with all 4. All right, we'll get started here. So basic stuff with ions. Any atom with a charge is referred to as an ion. If a neutral atom gains an electron, it becomes negative. This kind of ion is an anion. On the opposite side, if a neutral atom loses an electron, it becomes positive. This kind of ion is called cation. Amino acids are the building block of proteins. There are 20 different amino acids in the human body. We can only synthesize 12 of them. We ingest the other 8. The key elements, excuse me, the key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen, although other elements are found in the side chains of certain amino acids. Enzymes accelerate chemical reactions. The molecules upon which enzymes may act are called substrates, and the enzyme converts the substrates into different molecules known as products. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at fast enough rates to sustain life. Most enzymes are proteins, although a few are catalytic RNA molecules. These catalytic RNA molecules are called ribozymes. Enzymes' specificity comes from their unique three-dimensional structure. Like all catalysts, enzymes increase their reaction rate by lowering its activation energy. Some enzymes can make their conversion of substrate to product occur many millions of times faster. A covalent bond is a chemical link between 2 atoms or ions where the electron pairs are shared between them. A covalent bond may also be a term for a molecular bond. Covalent bonds form between 2 nonmetal atoms with identical or relatively close electronegativity values. The electron pairs that participate in a covalent bond are called bonding pairs or shared pairs. Typically, sharing bonding pairs allows each atom to achieve a stable outer electron shell, similar to that seen in noble gas atoms. If 2 atoms each share an electron equally, the bond is covalent. Many organic compounds, such as carbohydrates, result from covalent bonding. There is a covalent bond between the oxygen and each hydrogen in a water molecule. Each of the covalent bonds contains 2 electrons, one from a hydrogen atom and one from the oxygen atom. Both atoms share the electrons. A hydrogen molecule consists of 2 hydrogen atoms joined by a covalent bond. Each hydrogen atom needs 2 electrons to achieve a stable outer electron shell. The pair of electrons are attracted to the positive charge of both atomic nuclei, which hold the molecules together. And so then we'll also look here at polar and nonpolar covalent bonds. 2 important types of covalent bonds are nonpolar or pure covalent bonds and polar covalent bonds. Nonpolar bonds occur when atoms equally share electron pairs. Since only identical atoms, which have the same electronegativity as each other, truly engage in equal sharing, the definition is expanded to include covalent bonding between any atoms with an electronegativity difference less than 0.4. Examples of molecules with nonpolar bonds are hydrogen, nitrogen, and methane. A water molecule is an example of a polar covalent bond. The electrons are unequally shared, with the oxygen atom spending more time with the electrons than the hydrogen atoms. Since electrons spend more time with the oxygen atom, it carries a partial negative charge. Dipole-dipole interactions are a weaker form of polar covalent bonding. They are similar to hydrogen bonding, but carbon is more electronegative than hydrogen. Therefore, the magnitude of the dipoles are smaller. And from here, we'll jump to ionic bonding. Ionic bonding is the complete transfer of valence electrons between atoms. It is a type of chemical bond that generates two oppositely charged ions. In ionic bonds, the metal loses electrons to become a positively charged cation, whereas the nonmetal accepts those electrons to become a negatively charged anion. Ionic bonds require an electron donor, often a metal, and an electron acceptor, a nonmetal. Ionic bonding is observed because metals have few electrons in their outermost orbitals. By losing those electrons, these metals can achieve noble gas configuration and satisfy the octet rule. Nonmetals that have close to 8 electrons in their valence shells tend to readily accept electrons to achieve noble gas configuration. In ionic bonding, more than one electron can be donated or received to satisfy the octet rule. The charges on the anion and cation correspond to the number of electrons donated or received. In ionic bonds, the net charge of the compound must be 0. You can recognize ionic compounds because they consist of a metal bonded to a nonmetal. Ionic bonds form between two atoms that have different electronegativity values. Because the ability to attract electrons is so different between the atoms, it's like one atom donates its electron to the other atom in the chemical bond. Ionic bonds are interactions between charged atoms or molecules. The highly polarized nature of ionic molecules is reflected in their high melting point. An isomer is a molecule with the same molecular formula as another molecule, but with a different chemical structure. That is, isomers contain the same number of atoms of each element, but have different arrangements of their atoms. Butane and isobutane have the same number of carbon atoms and hydrogen atoms, so their molecular formulas are the same. However, each one has a different structural formula, which shows how the atoms are arranged. So, we can say that butane and isobutane are structural isomers. Okay, so again, just to make sure it's clear, structural isomers have a different structural formula. Geometric isomers, though, are two or more coordination compounds which contain the same number and types of atoms and bonds. So the connectivity between the atoms is the same, but which have a different spatial arrangement of the atoms. Isotopes are variants of a particular chemical element that differ in neutron number. All isotopes of a given element have the same number of protons in each atom. The meaning behind the name is that different isotopes of a single element occupy the same position on the periodic table. Monosaccharides are the most basic unit of carbohydrates. They are fundamental units of carbohydrates and cannot be further hydrolyzed to simpler compounds. Some monosaccharides have a sweet taste. Examples of monosaccharides include glucose, fructose, and galactose. Monosaccharides are the building blocks of disaccharides, such as sucrose and lactose, and polysaccharides, such as cellulose and starch. A steroid is an organic compound with 4 rings arranged in a specific molecular configuration. Examples include the dietary lipid cholesterol, the sex hormones estradiol and testosterone, and the anti-inflammatory drug dexamethasone. Steroids have 2 principal biological functions. Certain steroids, such as cholesterol, are important components of cell membranes, which alter membrane fluidity, and many steroids are signaling molecules which activate steroid hormone receptors. Muscles are composed of 2 major protein filaments, a thick filament composed of the protein myosin, and a thin filament composed of the protein actin. Muscle contraction occurs when these filaments slide over one another in a series of repetitive events. Collagen forms the connective framework of your muscles, bones, tendons, skin, and cartilage. Okay, here we have I know you just see energy, the law of conservation of energy. The law of conservation of energy states that the total energy of an isolated system remains constant. It is conserved over time. Energy can neither be created nor destroyed. Rather, it transforms from one form to another. And then we have our laws of thermodynamics. The zeroth law states that if 2 systems are in thermal equilibrium with a third system, then they are in thermal equilibrium with each other. This law helps to define the concept of temperature. First law states that when energy passes as work, as heat, or with matter into or out from a system, the system's internal energy changes in accord with the law of conservation of energy. Equivalently, perpetual motion machines of the first kind, which are machines that produce work with no energy input, are impossible. The second law states that in a natural thermodynamic process, the sum of the entropies of the interacting thermodynamic systems increases. Equivalently, perpetual motion machines of the second kind, machines that spontaneously convert thermal energy into mechanical work, are impossible. The third law states that the entropy of a system approaches a constant value as the temperature approaches absolute zero. With the exception of non-crystalline solids, which are glasses, the entropy of a system at absolute zero is typically close to zero and is equal to the natural logarithm of the product of the quantum ground states. Cofactors are mostly metal ions or coenzymes. They are both inorganic and organic chemicals that assist enzymes during the catalysis of reactions. Coenzymes are non-protein organic molecules that are mostly a type of vitamin. Looking at exergonic and endergonic reactions. In an exergonic reaction, the potential energy of the products is less than that of the reactants. Exergonic reactions are spontaneous. An example of exergonic reactions that occur in our body is cellular respiration, as shown by the formula. This reaction releases energy that is used for cell activities. Endergonic reactions require energy and therefore are not spontaneous. Photosynthesis is the classic example of an endergonic reaction. In an endergonic reaction, the change in G is greater than zero, which means it's positive, while in an exergonic reaction, the change is less than zero, which means negative. An active site is where the enzyme and substrate bond. A substrate is the molecule an enzyme is working on. Activation energy is just the energy required to begin a reaction. Induced fit is the substrate getting the enzyme to alter its shape so that the two can bond. This leads to an enzyme-substrate complex. Once a substrate enters the active site of an enzyme, the substrate gets the enzyme to alter its shape so that the two can bond. That's active sites. And then finally, we have competitive inhibition. In competitive inhibition, there are 2 different substrates that compete for an active site on one enzyme. Both substrates cannot bond to the enzyme. The substrate in greater concentration will most likely bond. A non-competitive inhibitor is a substance that interacts with the enzyme, but usually not at the active site. The effect of a non-competitive inhibitor is to change the shape of the enzyme and thus the active site so that the substrate can no longer interact with the enzyme to give a reaction. When an inhibitor binds to an allosteric site, the active site changes shape while the substrate is unable to bind to the enzyme. Okay, and then finally we get back to our questions. So this is a great point to go ahead and hit that pause button and see how you do on answering these 5 questions.